Hi, my name is Maria Gomez. I am a graduate student at the University of Minnesota Duluth, and I study plant population genetics. So I pretty much look at the DNA of different populations of trees to identify differences between them. So today I'm going to share with you how to do a DNA extraction and how a little bit about how DNA works. DNA is my favorite molecule, so I'm really excited to share all this with you. So all living organisms are composed of cells, right? And inside each cell, we can find DNA. DNA stands for deoxyribonucleic acid and has a double helix shape. This molecule carries the genetic code for all living organisms. So think about it as a recipe that determines what a tree or a dog or a bird or human should look like. The cool thing is that within every cell we have a copy of our DNA. And we can use that DNA, that information, to find difference between species, between populations. We can see where different organisms come from, for example. We can also understand how different organisms have adapted to their current environment. And we can even recreate phylogenies, which are like family trees that can go back to many years. And this is possible because even though DNA is unique to every organism, the genes, which are like Legos, think about them as like building blocks, are very similar across organisms. And also because organisms receive half of the information from each of their parents. Receive half of the information from one parent and half of the information from the other parent. So if you think about this, we can really get a lot of information from the present and the past of the organisms. And we can answer, as you can see, so many questions using this molecule that is DNA. And in order to do that, we have to usually extract DNA. And then we use some high technology to really get the sequence and find those differences. But the first and crucial step is to really get the raw material, which is the DNA molecule. And we actually can do that with materials that we can find around our houses. So I'm super excited to show you today how to do a DNA extraction from your own cheek cells. Get started with our DNA isolation. For that, you're going to need the guide that you received in your kit. You're going to need a medicine cup, a plastic transfer pipette, which looks like this. You're going to need one 50 milliliter tube, which is the big one with the red cap. You're going to need one 15 milliliter tube, which is the blue one. You're going to need your tube that has the rubbing alcohol, which is the other blue one that has the label to not consume. You're going to need a 15 milliliter tube with salt. This microcentrifuge tube, and a microcentrifuge tube is this small tube that has dish soap, and it should be labeled DS. And then the other small tube, the microcentrifuge tube with food coloring, labeled FC. And you're going to need the paper clip. From your home, you're going to need a cup, some water, and then you can have a container with snow or with ice. I think snow really helps keep everything really cold. So go outside and get some snow. Okay, so let's get started then. So the first step is to prepare the blue rubbing alcohol. So you're going to take, with your pipette, 3 drops of your food coloring, and you're going to add it into the tube that has the rubbing alcohol. And you should mix it very well, and that should give you this blue rubbing alcohol. And we're going to set that aside for a second. Now we're going to prepare that ice bath. Just make sure that there's enough either snow or water to really cover the tube. We're going to take the rubbing alcohol that we made, and we're going to put this tube in in our snow bath. Make sure that it's nice and chill and well and all covered. That's our second step. Our third step is to prepare the DNA isolation tube. So you're going to take the small microcentrifuge tube labeled dish soap, the clear small one with dish soap, and you're going to pour the dish soap into the 15 milliliter conical tube, like the empty one, and you're going to put that in our ice bath with snow or with ice. So we're going to pour it here. Make sure that everything is there. You can give it a little tap if it's sticking to the sides. Just be patient. All right, once that is all out, close that. We're gonna cap this tube with the soap and we're gonna put it back in our ice bath again. Again, make sure that it's all covered and staying cold. All right, we're gonna go into step 4, and step 4 is to prepare a 0.9 saline solution. So saline solution means that a solution with salt. Take our tube that says salt, and we're gonna take about 2 cups of water, which I have right here, and we're gonna combine the salt with the water. Just gonna pour it right in. Don't get rid of this tube because this will help us later measure some volumes. And we're just gonna stir it and make sure that all the salt is dissolved. An important thing while I mix this is to make sure that you're using the right amount of water. It's the right volume of water. So make sure that you have 500 milliliters, which is around 2 cups of water. So now that my salt is dissolved in the water, I'm going to take the 15 ml tube that I used, and I'm going to measure 10 milliliters of that solution and pour it into the medicine cup. So you can literally kind of just scoop it up. Right. So 10 mils would be up to the 10 line. So make sure that you have the right amount. Just here. All right. So right now I have 10 milliliters of our 0.9 saline solution. And I'm going to pour this into our medicine cup. You can also use your medicine cup to measure, but I think that the tube is easier to really know where the 10 mils are. So now we're going to take our saline solution and we're going to sip it. Don't drink it, just hold it in your mouth and just swish the solution around your mouth for 30 seconds. Alright, so after the 30 seconds, you're going to spit all the solution back into the medicine cup. Now you're going to take your medicine cup and your conical tube with the dish soap, the one that was in the ice with the dish soap, and you're going to pour your spit slowly. Alright, so when we did that wash in our mouth, pretty much we are getting cheek cells into that spit solution that we had. And now when we add it to the dish soap, what we're doing is we're breaking down the plasma membrane, so the membrane around the cell, and releasing the DNA from proteins inside the cells. We're going to cap it very well, and we're going to gently rock the tube lengthwise back and forth for 2 to 3 minutes. Don't do it too hard, because if you do it too hard, then you're going to break the DNA molecules, because DNA, it's fragile, it's a small molecule. And we're going to get shorter pieces, and we're not going to be able really to extract it and see it in our tube. So literally just rock it back and forth very gently for a couple minutes. You can have another mini dance party like we just had when we were doing the wash. So remember that right now we're breaking down the membrane of the cell and releasing the DNA into our solution. Right, so just keep rocking. We're going to actually isolate now the DNA. So take out everything that is not DNA that was part of the cell out and separate those. And the first thing is we're going to separate those bubbles from the DNA sample. So what we're going to do is we're going to take our DNA sample and we're going to pour slowly most of the DNA sample into the big 50 milliliter conical tube. So what we want to make sure is that all these bubbles that we got at the top, we kind of get rid of those by just slowly pouring it into the red tube, because our bubbles are going to go to the bottom of the tube. So pour it slowly, and if you start seeing that you're starting to get some bubbles, just stop before you drop the bubbles. So, okay, I'm getting some bubbles right there. So you can see everything that is left right here is mostly just bubbles. And in this tube, I have a solution with no bubbles. All right, so I'm gonna cap my tube again. So now that we have this tube, we're also going to get our tube with our alcohol. So, okay. So we're going to take our tube right there, and we're going to take this blue rubbing alcohol, and we're just going to tilt this one very gently, and we're going to add the rubbing alcohol very slowly, trying not to disrupt our cells. Let's not disrupt our DNA. So very gently. All right. So now that we've added the alcohol, our DNA is going to start to solidify, and you're going to start seeing some white threads at the edge of the ethanol and the DNA sample after a minute or so. So just give it some time, and we'll look at the results in a couple seconds. So we've let our DNA sample sit for a minute. You should already start seeing some white threads kind of by the boundary between the ethanol, the blue ethanol, and the DNA sample. So now we're going to straighten a paper clip, and we're going to use this to collect our DNA. Okay, so I'm actually going to leave a little edge so that it's easier for me to actually hold it and twirl it. So you're going to take your tube, you're going to open it, and to collect our DNA, we're just going to go inside and just swirl really slowly inside our tube. So I have some here, so I'm going to show you. Hopefully you can see. So do you see those blue kind of threads around the paper clip? That's our DNA. So keep twirling, see if you can get a little more. But yeah, pretty much there you have it. You've just extracted your own DNA from your cheek cells using a lot of very simple ingredients that you can find at home. And I want to just very quickly kind of explain how you can really use this. As I said in the beginning, you can use this to really look at how related different organisms are. In my case, I use it to study tree genetics, right? So I want to see how different are the trees. I look at specifically oak trees, so how these trees that are grown in Duluth, for example, that were really close to the lake, are different from trees that are grown in, let's say, the Twin Cities, right? Which is a little bit far away, right? So why do we have genetic differences between these two organisms? Are they due to the environment, or because they're just too far away? And we can answer these questions by extracting DNA from the trees in very similar ways as we did today, and then looking at that actual DNA sequences and finding differences between them. So that's pretty much it. I hope you guys had a lot of fun. I certainly did. I hope you were fascinated by DNA just as I am. DNA is one of my favorite, if not my favorite molecule, because it's so cool, because thanks to DNA, we have all the different organisms and living things that we have around us. So hopefully you saw a little bit of the process of extracting DNA and can see how cool this molecule is, and how in just a small tube like this we can actually have so much information about our organism. So if you have any questions or any comments, or if you want to show me your results, let me know and I'll be happy to chat with you.